FORA.tv. The world is thinking.
In the 1850s, Pasteur demonstrated that living microbes were responsible for the chemical transformations that occur in fermentation. And he did that with an applied project, which I also, you'll notice I'm saying a few things about science as I sort of go through this, that often we make these sort of big distinctions between basic science and applied science. We should be careful about that, because quite often thinking about applied problems can be really very relevant to fundamental understanding. And in Lille, in the 1850s, he was asked by the industrialist responsible for beet sugar fermentation to investigate why sometimes, instead of making alcohol, they went sour and made lactic acid. And what he discovered, simply by looking under a microscope, I should say, is that when you made alcohol, yeast was discovered in the fermentation, and when you made lactic acid, you found bacteria in the fermentation. So he simply argued and advised the industrialists that just make sure you have plenty of yeast there and everything will be fine. And that indeed led to much more reliable fermentation. But he went one step further. He said, and this is translated, fermentation was a physiological act yielding chemical products necessary for the cell. In other words, such chemical reactions are an expression of the life of the cell. What he was stating there is that the life of the cell can be understood in terms of chemistry. And that was really the basis of life as chemistry. And by the turn of the 20th century, it could be argued that life's processes could be reduced to chemistry. That led to the birth of the science of biochemistry. We have a graduate program in biochemistry, I was hearing in the introduction. And Loeb from Rockefeller University stated that the cell is a chemical machine. The cell is a chemical machine.